So today I would like to start, talking about muscles and the biomechanics of a human skeletal muscle. And that's lecture 9 on D2L, the one that you have. So we're going to talk about the basic behavior properties of the musculotendinous units and how skeletal muscle function to produce coordinate movement. And then we'll talk about the important parts of force velocity, length tension, and electromechanical delay. Right? These are the things that we're going to cover. Now, here's the thing. When I cover muscles or bone or any of these topics, what I like to do is to talk about the general anatomy of the muscle, a quick review, and then talk about the problems that we face with muscles, and then what kind of technologies we can use to solve these problems. Does it make sense? So quickly, if we go into the next slide here, see these pictures? You can see a normal resting length muscle that's on the top, extended if it's stretched, and you have the after passive elastic recoil. And what do you mean by passive? Someone, when you do passive stretching, it's someone is doing it for the subject, right? Now, contraction, let's say this is the biceps. When we do contraction, the biceps will get shorter and bulkier. Okay, now, as engineers, we need to be able to measure quantifiable parameters. Remember, math does not lie. So what we need to do is to find things that we can repeatedly measure and quantify. So one of these parameters is something called extensibility, which is the ability to be stretched or to increase in length. Now, as engineers, how can you measure this? How can we measure extensibility without having, the muscle to be damaged? What would you do to measure extensibility? Say it again. Ranges of motion. That's a very good one. And how can we use? We can use the goniometer, or we can use. So extensibility is one of the things that we can look at is actually the range of motion. And you can use the goniometer, or you can use videos and Kinovia to measure those. Good. And as I told you, it could be good or bad if you have low extensibility or high extensibility, but it depends. Now, what about elasticity? Elasticity is the ability to return to normal resting length following a stretch, right? That's another. Here's the thing. I want you to think about giving quantifications or numbers to each one of those. So extensibility, range of motion, that's good. The elasticity is the ability to return. Here's an important aspect. When you stretch any muscle, it's not that it goes back to its normal length. The important thing is how quickly it goes back to its normal, which is something we call recovery. Does that make sense? The recovery thing. And able to return to normal resting length following a stretch. It's tricky in how do you think we do measure this using technology? If someone asks you, how would you measure elasticity of a muscle non-invasively? And remember, means that we're going back to normal resting length, correct? So how would you measure this? What would you use technology to measure? So you can say that when you do excitation to the muscle, you have high activity of the EMG, but when you rest it and it comes back to normal baseline, that's when it goes back to its resting length. So, extensibility and elasticity, and I'm glad that you all are thinking about this way. Now, we need to think about muscles. I know that you covered this in anatomy and physiology, but here's what I want you to think about. We would like to try. A good engineer is an engineer who can simplify an organ or an anatomical structure, right? Now, what do we mean by simplification? Simplification means that you are trying to use known components in order to represent a biological tissue. Now, what are the things that we continuously know from physics? Resistance, correct? And a contractile component like a piston, a battery, a capacitor. So all these elements from mechanical and electrical engineering, we can use them to simplify biological tissue. Now, why do you think we want to use them? Because we know how the resistance works and we can predict its functionality in the future, right? So that's why we would like to look at the muscle now and see if someone will ask you, can you model or simplify muscles? What would you represent them with? What would you say the muscle can be represented with? That's bone. Levers are bone because the muscle itself is something that is stretching, right? Is pulling. So one of the things that we can look at the muscles is elasticity, like a rubber band, an elastic component. What would you represent something that is elastic in electrical engineering? A resistance, right? Because it resists elongation, right? It's the same thing, resistance. And what Alyssa mentioned about the muscles pulling the bone, it's a contractile component, just like a battery. Something is giving you energy, right? Or force. Electrical engineering we call it electromotive force. But here's the thing. So we have components of the muscles, muscle elasticity. You talked about elasticity and looking at how it goes back to its normal size. But what is the thing that is pulling it back to normal size? So what we do is this. We say we have two things. We have something called parallel elastic component, and we have something called series elastic component. Now, the parallel elastic component, it's passive elastic property of muscle derived from muscle membranes, right? So muscles, from your anatomy, muscles are made of fibers, correct? Each muscle fiber is one cell, and these cells are very long. They could be the length of your muscle, and they're multinuclei. Now, the membrane of these fibers, of these muscle cells, is actually what's giving the parallel elastic component, the elasticity. And the series elastic component is the passive elastic property of muscle derived from tendons when a tendon muscle is stretched. So we have two things adding to elasticity, which help muscles go back to their normal size, right? So you have the tendon and you have the membrane. Do you remember what membranes are made of? Phospholipid bilayers or cell, right? It's a cell. The fiber of the muscle is a cell. So these phospholipid membranes can add to the PEC, and the tendons themselves, these tendons will add to your SEC, which is the series elastic component. Now, remember, a good engineer is the one who can simplify a system. Now, we're looking at biological systems, and that's the difference between us and mechanical engineering or electrical engineering. You need to be able to represent a biological tissue using predictable component. We can predict component that you can predict their functionality. That's the most important. So I'm not going to discuss anatomy and physiology. You covered that. But I want you to think about elasticity and resistance and contractile, forces that can create the inner force. Does that make sense? We good? Perfect. Now, so this is the schematic representation, the modeling. Remember, modeling is representing a biological tissue with a, components that we can predict their functionality. So from mechanical perspective, musculotendinous, because this is muscle and the tendons together as one system. Remember when we talked about biomechanics that you can select different parts to be what you're studying? I'm studying the muscle and the tendon together. So the musculotendinous unit, that's what we call it, behave as a contractile component. That's your contractile component, like a piston or something mechanical. And then the muscle fibers, which, remember we have the actin and myosin, and they're sliding on each other in the muscle. That's what's creating the contraction. But you have things that want to get you back to the normal length, which is the resistance, the elasticity. And that comes from one elastic component, which is the muscle membrane, and the series one, which is another elastic component comes from the tendon. So that's the series, that's from tendon, because it is stretching muscles. Remember when we talked about the Golgi body yesterday, on Monday, that is trying to relax the muscle instead of just keeping it contracting? That's what's happening here, and the parallel are the fibers themselves. So what I want from you, it's an exercise. It's not just muscles. Any biological tissue. Can you think about what are the components that you can represent that tissue with, that you would predict them, and you know how? Does it make sense? Good? Okay. Now, a quick thing about muscle activity, the musculotendinous unit. Remember that muscle contracts because of action potential that comes from the brain, which are the neurons, right? And we know that we have the peripheral nerves that comes out from the spinal cord, correct? And they go to control the muscles, the motor nerves, and you have sensory nerves that are coming back. So you have something called afferent and efferent pathways for the muscles. Now, the point that we would like to make is that what can we do as engineers, or what kind of technology we can use to study that behavior, which is exciting the muscles. So in the previous slides, we talked about contraction. We assume that the muscles are active. Now, how would you study this connection between the nerve and to try to simplify it and to measure, quantify things? Remember, we got into engineering because we'd like to compare quantifiable measures. So we can look at something called irritability. That's another parameter, and it's the ability to respond to stimulus. How can we measure irritability? It's EMG, but you need to measure two things. You need to measure the EEG, the brain activity, and EMG at the same time, and put them together on the same graph and see how fast or slow the muscles will contract responding to a stimulus from the brain, right? Now, that's one way. Another way of doing this is looking at, you remember when what do you call that test when you have like a small hammer on the knee? The reflex test. That is a measure of irritability because it gives you how quickly the muscles are responding to stimulating the nerves, right? Because you're stimulating it locally in there. So yeah, EMG, EEG, using some mechanical loads on specific locations in the body, that could be something for you. So, and also what Alyssa mentioned is that we are measuring this non-invasively, right, on the surface. But you can have also electrodes in the body that's invasive, and you can put these electrodes to measure from the activity of the peripheral nerves, and then measure the EMG and find the speed and the between them. So you can have it invasive or non-invasive ways to do that. Okay, now ability to develop tension. It's the contractile component of muscle function. How would you measure develop tension? Is force. So you can measure. How would you measure how much force your muscles can handle? You can use dumbbells, right, and lift them multiple weights, and you see them. And we're going to talk about something called, have you heard of something called eccentric or concentric muscles? So you can have your muscle with its original length and hold weights and see what is the weight that you can hold without changing the length of that muscle. And we're going to talk about that. Here's what I want from you to think. What are the things that we can actually measure and quantify with repeatability, and we can predict what is going to happen as out if you modeled it as a okay. So all the terms in red are things that we need to be able to measure using okay. Next, now this is another important exercise that you would like to be able to measure using sensors, correct? And we're going to do an exercise. I want to do exercise with you about sensors. But before we start, you know that we have the spinal cord, and we have two pathways. There are neurons coming from, or nerves coming from the brain, and you have sending signal from the brain down, and the other side is taking signals, the sensory signals, back to the brain. So that's why we have this compartmental shape of the vertebrae, right? Or the spine itself. So blue coming from the brain, the brown is going back to the brain. That's the efferent is going down, correct, from the brain to the muscle. The afferent is the thing that going up to the brain. So the blue is efferent and the brown is afferent. And now here's the thing: we can stimulate muscles using electrical activity. We don't recommend it, but you can do it, right? So I can see this blue now here. So I can design a device that can stimulate muscles, and then the muscles will send sensory feedback to the brain and do the whole thing. Now, I know that a lot of you play sports. Have any of you used any electrical stimulation? Right? It's good and bad and scary at the same time, because if there is any fault in this, it can actually do micro shocks in the body, and that will stop the heart. It's bad. So you need to be very careful with this. I don't know how big of a size of that thing and how much voltage or how much current, but it could actually create problems. People need to be careful, very careful with this. Stimulators. This could be a study actually, and to study the extensibility and flexibility and EMG after using that stimulation. Does it make any difference, or what is the difference that's making? So that could be a research study actually to do. I would be interested in looking sometime. But it makes sense that because muscles act actually based on electrical stimulation. Now, do you know what are the important things in the electrical stimulation? It's the amplitude and the frequency, right? How much of a voltage or current and how frequent the frequency of that stimulation. Your muscle activity on the surface, the electrical activity, will be measured by this device, the microcontroller. And depending on how many muscle fibers you're contracting, the light will you have. And the things that you're going to be building these next semester. Right. So this is what we're doing here. These sensors actually, you might be measuring the muscle activity, but instead of the strength, you're measuring the electrical activity of the muscle. And you're going to be soldering this and building it in the instrumentation class, and then using it to control the robotic arm. And I have multiple of these sensors for other muscles to collect. Okay, so does it make sense? Are we good? Are we good with what we are planning on doing? This is measuring. Now, the thing that you told me, the stems are giving the signal to the muscles to contract. Now, if you are not doing it correctly, it could be dangerous. So you need to be careful actually with dealing with that. Okay, so I have these kits for you when we do the instrumentation to build them, and they come actually something like this. See this? That's how you'll be getting them, and then you will build that. Okay, we good? Okay. Again, what is our goal? Our goal is to find quantifiable measures and then use this information into something useful. Now, do you remember the muscle fibers? So again, each muscle cell, we call it muscle fiber. And each of these fibers actually, it's a multinucleide, very long cell for that. This is a scanning electron microscopy image of it. A single muscle cell surrounded by a membrane called the sarcolemma. Is that how you pronounce it? Sarcolemma. Now, this is what is giving you you remember the resistance, the parallel resistance, and the series resistance is coming. The elasticity is coming from the tendons themselves. Okay, and we have inside the cells. I don't want to go into the anatomy of it. I think you should. Sarcoplasm, which is the liquid inside these cells, and that's how they look like. The muscles. So what do we know about muscle fibers in general? Not the anatomy, but in general. The things that we know is that some fibers run entire length of the muscle. So I think the longest one are the muscles in the thigh, right? It goes the entire length of the thigh. That's a very long muscle. For one cell. Others are shorter. Skeletal muscle fibers grow both in length and diameter from birth till adulthood. So they continuously grow. Now this is different than cartilage and nerves. So that's another important aspect. So how would you assess if someone wants from you to look at the length of a muscle fiber? Can we do it non-invasively? This is a big question actually. Can you assess the length of fibers in a specific muscle for a specific person? Non-invasively. That's a big challenge actually. And the best way to approach this is through imaging. Colored imaging actually. You need to color the muscle fibers to see that. Growth in length and diameter. We know that it grows, but we don't know how much and what kind of problems we can face with any medical condition that will affect the growth of muscle fibers. Something to think about. Fiber diameter can be increased through resistance training, and that's something important. When you do resistance training, actually what you do is you're increasing the size of the fibers. Does that make sense? How we do it? Now, a bigger fiber, what does a bigger fiber mean? What is the main internal structures of muscle cells? So if the cell is getting bigger, what does that mean? The diameter of that cell. So a bigger muscle means that you have a lot of proteins in it. Proteins mean that you have more fibers, actin and filament and collagen, right? Because that's what does mean that have a bigger muscle, a bigger fiber. So you have a lot of proteins actually inside that muscle fiber. Still, it's something. I'm not an expert in muscles, but I think it's something people are trying to assess and optimize. Okay, you remember this? The myosin and actin, and you know that the thin filaments are the actin filaments, right? And the thick one are the myosin, and the myosin arms move to bring the actin filament together, and that's how contraction takes place in the muscle. This is something you studied, the H band and the A band and the I band. So I'm not going to go into this, but what is important is understanding the relationship between length and strength. Does that make sense? The length of muscle and the strength of muscle. And do you think that if you have so we are contracting the biceps. Do you think that the shorter the biceps, the stronger mean that it's going to create stronger force is going to create? We have a biphasic shape, because if you have, let me show you something. Let me draw something for you. I'm going to remind you of these, the bands. Those are the myosin, those are the actin, and how you do the contraction with them. You can check those. This is a scanning electron schematic of one muscle fiber. See, it's a very long fiber. It has multiple nucleuses and multiple nuclei, and then you have this contractile microfibrils in it. You know about this. Now, this is the structure between bone and the tendon, right? Remember, we have something on the bone called the periosteum, which is the outer shell of the bone, and that's where the tendons are coming out actually from it, like a branch. Now, what is the definition of a motor unit? Now, here's something you might not covered it before. A motor unit is one motor neuron with all the fibers that it touches. So if you look at this image here, see this? This is spinal cord, right? We have a motor neuron coming out from the spinal cord. Now, this motor neuron has its body, right, inside the spine, but it has the, the axon of it. Bless you. The axon. Do you know what are the things on the axon? The small brownish things, stuff, cells. Those are called Schwann cells. Do you know why they are there? The speed, because the action potential will jump between them, between the spaces, and that speeds the communication between the brain and the muscles. Correct. Now, this is the important part. See this here? See those dendrites? Just like roots of a plant, each one of them is going to touch only one muscle fiber, one muscle cell. Does that make sense? Now, each one of us, we have different combinations of that. So when I tell you a muscle fiber, a motor unit, what does that mean? This nerve, right? Motor nerve, with its dendrites and all the muscle fibers it touches, it's called a motor unit. I might have motor units that have, let's say each one neuron could have 10,000 muscle fibers. You could have 50,000. Someone could have 20,000. So each one of and this is important because that affects how do you excite your muscles, how quickly the excitation is there. And that's a genetic issue. That's how many muscle fibers are connected to each motor unit, motor neuron to create a motor unit. Are we good? So a motor unit is a motor neuron with all its fibers, muscle fibers that motor neuron is touching. Now, single motor neurons and all fibers, muscle cells, it innervates. That's called a motor unit, and it's considered the functional unit of the neuromuscular system. So if you'd like to say what's the performance of a specific neuromuscular activity, we need to look at how many motor units we do have. Now, here's the thing. Here's the cool thing. Imagine if someone gets a spinal cord injury, right? So what does that mean? It means that we have damage in the spine, and the motor neurons are not acting anymore. If I remember, we have efferent and afferent pathways, correct? We have things coming down to the muscles and things coming up from the muscles to the brain. Now, if someone is paralyzed, but we kept doing exercising for his leg or her leg, the body is going to reprogram the nerves in the spine to find another pathways to excite them. That's why it's very important to continuously keep doing exercises for people with spinal cord injuries. And that's an engineering problem. We can't have physical therapists all the day with someone, but you can build a robotic system that can do a movement for someone to exercise. The more you do exercise, the more actually we do rewiring and reprogramming for the pathways. Consider this as a highway going back both ways. There's a lot of cells here, nerves. If some of them are damaged, we can actually reprogram the spine to move on another pathway, to take another pathway, as long as we don't have a complete cut in the spine. All right, so what are the things that are important? Here's the first graph that we would like to look at. We have something called fast twitch fibers. They reach peak tension and relax more quickly than something called slow twitching fibers. Remember, fibers, what fibers mean? One cell. A muscle fiber is one cell, one muscle cell. We don't call them cells, we call them fibers because they're very long. So the fast twitching fibers reach their peak tension. So this is fast twitching, right? See how quickly, that's time and that's tension. And the experiments that have been done is that we take the fibers outside of the body, put them in a liquid, change the concentration of minerals, calcium or potassium, and see how they're going to contract. And on both sides, we have some sensors to give us the force, the tension. So fast twitching, they do the tension, which is actin and myosin contraction, and they relax quickly, and then the slow twitching fibers are the ones that they take over. Here's the question. How is this useful for us? Or as an engineer, how could this be useful clinically? You can study the effect of drugs on this, right? You can study the effect of, frequent exercises, and to see what is happening with the fast and the slow twitching fibers in the muscle. But I personally like to look at the effects of drugs, or, anything that we eat, that is going to, how would that be affecting our fast or slow twitching? What are the quantities that you can measure here? First thing is finding the maximum force, right? Tension, correct? And the time to reach the peak tension, here and there. So you can report these two values, and you can compare, let's say, the effect of a specific drug on these two values. And then for the slow twitching, you know the peak is going to be similar, close, but the time is going to be completely different. So if this is 5 seconds, this is going to be 10 seconds, right? So the quantities that we can quantify are the tension, maximum tension, and the time. Okay, so we have characteristics of type 1, slow twitching, and we have type 2, fast twitching, and we have type 2B, fast twitching, glycolytic. So oxidative means that you have oxygen, right? You have a lot of oxygen. Glycolytic, it could be that the energy is coming mostly from using glucose to get ATPs, right? So you can look at these, the contractile speed for each fiber, the fibers, what happened with them. If that is a slow twitch or it is a fast twitch, we have two types of them, and what are the characteristics of these fibers to look at them. So if I told you I have type 2B fast twitching, the contractile speed will be fast, the fatigue rate will be fast. How would you characterize fatigue in muscles other than damage? How would you say that your muscle is fatigued? What does that mean? Because that's correct, but why? It's the lack of oxygen and the accumulation of CO2. If you are, our muscles store oxygen, but if you're continuously using it, it depletes the oxygen and it has a lot of CO2 accumulating, and that is going to stop. It will continuously be contracted. It will stop being relaxed, and that's where injuries actually take place, because now you're damaging, because everything is contracted and you feel that your muscle is stiff. So what does that mean? Stiff means that it's contracting, but it's not going back to its normal. It's not relaxing. And then it gives you an indication of the diameters, how much ATPs the concentration of needed ATPs. And remember the Krebs cycle, converting glucose to ATPs, and the role of the mitochondria and all of that thing. These are all biology. I'm not going to go into this, but there's something to look at, to consider. Now, structural organization of the muscle. How are muscle fiber organized? So we have parallel fiber arrangement, and we have pinnate fiber arrangement, one for the long and one for the short. So if you look at this image here, so if it's parallel fiber arrangement, it is the fibers are parallel to the longitudinal axis of the muscle, right? If it's pinnate, shorter fibers are attached to one or more tendons, and you can look at it. So this is parallel, right? What do you call this muscle? The abs. What do you call those? Yeah, they have. Yeah. So these are parallel. Those are the pinnate. Unipinnate is in one direction. Bipinnate, you have two directions for the muscle fibers, and multipinnate have multiple directions. And that's from your anatomy. Okay. Now we have the angle of pinnation. If we are able to do invasive measurements or imaging, for that, you can look at that the angle increases as tension increases in the muscle fibers. So that is something to look at, right? Relaxed or in tension. So look at that. The angle becomes larger, right? Okay, skeletal muscle function. What is the most important function of our muscle? It's the thing that, work or, move the levers, right? The bones. So torque produced by muscles at the joint center, or rotation is produced by muscle force, and you multiply it by the distance. So we talked about this. We're going to have more about it. Now, here's the thing. How are motor units recruited? If you have slow twitching, motor units are easier to activate than fast motor twitching, because they require less energy. And the slow twitching motor units are the first to be activated, right? And as the force requirements, speed requirements increase, or activity increase, the motor units with the high threshold are progressively activated. So that's another thing to keep in mind, actually, that slow twitching fibers are activated first. Where can we see this? When you do the EMG measurement, remember when we look at the lights from Hayden, right? So the first one to be activated are actually the slow twitching fibers, and then we have the fast twitching fibers. Okay, do you know that? So if you have your muscle on its own length and you're holding a weight, that is called isometric concentration, right? Sorry, isometric contraction. I don't know where concentration, from ATP, so sorry. So isometric contraction. And what that means that you are holding a weight, but the muscle is on its same length. A concentric contraction if the muscle is shortened, and eccentric contraction if the muscle is elongated actually, and you're holding a weight. So let's say if I have one of these, the muscles that we group muscles that affect the knee. Okay, so where is this important for us? We can do some, and before we do this, are we good with agonist and antagonist muscles in doing the movement? There is something called neutralizer. It acts to eliminate an unwanted action. And we have something called stabilizer. Muscles act to stabilize a body part against one another. The agonist is the one that causes the movement, and the antagonist is the one that tries to slow or stop the movement. And then you have stabilizers and immobilizers. Shoulder abduction. What is shoulder abduction? Adduction is this way, right? That's abduction. So if you're doing shoulder abduction, so the deltoid is the agonist. Which one is the deltoid? It should be the agonist. It's the one that's moving this way, right? The antagonist is the dorsi, the latissimus medius dorsi. Which one of those? Yeah, one of the activities that we will be doing is when we do the, let's say, squatting or jumping for basketball, which is the agonist, the antagonist, stabilizer, and neutralizer muscles when you do these activities, and then look at their activity with using the EMG activation. Okay, so that's good. Now we have something called what are the disadvantages associated with the function of two joint and or multi-joint muscles. Active insufficiency and passive insufficiency, that is related to how many muscles should be acting when you're doing a specific motor task. Now, remember, the shoulder abduction is a standard movement, but that's actually not what we do usually. You need to have other things moving, in different. Nobody's walking, every day doing this, right? But this is a standard movement that we would like to study. Okay, skeletal muscle function, passive insufficiency. It's the inability of a two-joint muscle to stretch enough to allow a full range of motion at both joints, and that's related to the anatomy or the structure of the body. So two joint muscles to stretch enough to allow a full range of motion for the joint. For example, flex the forearm. How do you flex? Let's flex the forearm and flex the fingers at the same time, right? You're not going to get your hand all the way down. So if you do this, try to do this, you're not going to do, if you don't flex the fingers, you can go down, right? And that's a limiting factor. And people who know martial arts, they can leverage this in hurting other people actually, and with dislocating the joints. How to do that? That's passive insufficiency. Decrease the range of motion of the wrist extension with the finger extended, right? So if you do this, right? If extended, they can do this. But close this and try to do that. You're not going to, so do this all the way back. Close it. Try to go back. You don't go to all the range of motion. And that's actually how people try when they do, anything. You want to move, you want to control someone, you need to understand these limitations. And then you can control their posture. Now, here's the thing. You can study this range of motion, and also you can study the EMG activity. And that can give you the indication about the health of the muscles. So if you do this, you can go all the way down. If you close this, you can't go down. Now, you can do this on the other side, but if you close it, you can't go all the way back, the muscles. Yeah, see how big the angle here compared to this angle? So close, try, going to go all the way down with this, with the joint. And those we call them two joint muscle to stretch. Okay. Active insufficiency condition occurring when a two-joint muscle cannot shorten enough to cause a full range of motion both joints. Because, see, remember, the fingers are joints, and also the wrist is a joint. That's how we're looking at this. Decrease, wrist flexure, that's the same thing. Passive and active. Active means that you are using a lot of muscles to get you down there. Okay, I will stop here, because the other part is going to be looking at the force, velocity, length, tension, and the electromechanical delay. Again, I don't want to repeat your knowledge about anatomy and physiology, but we would like to think about technology. What are the things that we can do to measure quantifiable parameters related to muscles? Does it make sense? That's the point. Can we do things for our muscles, and what are these things that we can do? I know, one thing quickly before we have 5 more minutes. Something quick, just quick. about the force velocity. See this image here? So this is an important force velocity relationship graph. And I would like, if you're an athlete, you should be building this for yourself. Seriously. So, and here's what's going to happen. You need to get weights and get a series of weights, a large number of different weights. And what you want to do is, let's say for you identify that this is your original length of the muscle, right? Here's what you need to have. Keep adding weights to your hand, holding them, until you reach the point where the weight is moving. So that the x-axis is velocity, right? You need to find this force. See this force, this point here, this point? You need to see how much you can handle by keeping that muscle with its original length. That force is important because that's going to identify your eccentric and concentric and the isometric. This is called isometric maximum. That's the maximum weight that you can hold, keeping your muscle length the same, right? Now, the problem with people who get ACL tears and all the other injuries is that they are exceeding that limit while trying to be static. You can't have the zero velocity and have that much weight. And that's what happened when they try to stop while they're running and they try to stop. They try to go to zero weight, but they have a lot of force in that direction. And that's what creates the tear. Does that make sense? Then, what happened here on this red box, look at this. You have the maximum force going up, right? You have the maximum force, zero velocity. Does that make sense? Now, if you increase the velocity and reduce the force, what is this? Do you know when you do what do you call lifting weights? This where you supine position and lifting weights. What do you call it? Bench press. And you have the weight. Now, if you're going down, the velocity is increasing, but the force is going to zero. So where is that here, right? Because it goes down quickly to the chest. If you're pushing it on the other side, what are you going to do? You're increase the load, the force on the muscle, but it's hard for you. The speed will go smaller and smaller, right? So you need to think about these eccentric and concentric muscle movement. Whatever you're doing in playing sports, you need to understand if you are performing the motor task with a concentric or a eccentric muscle. That's important because that could damage actually your muscle without knowing what's happening with it. And this point, no matter what sport you need or find the most used muscle, you need to find this isometric maximum force that you can handle at a zero speed with the same length of the muscle. That's important. And we will do this. We're going to do a couple of exercises for the knee because that's what we're interested in actually studying now. Does it make sense? We good? Yeah. It's not the anatomy. What's the technology that we can use to get into the whole thing? Thank you, guys.